Okay, I think many people are now safely back, and so I think I will launch our second speaker tonight. So the second speaker is Professor Lisa Feldman Barrett. Lisa is a university distinguished professor of psychology at Northeastern. She has appointments at Harvard Medical School and at Massachusetts General Hospital. As if that wasn't enough, she's also Chief Science Officer for the Center for Law, Brain and Behavior at Harvard University. Lisa is one of the most cited scientists in the world, and her new book, 7.5 Lessons About the Brain, is launching with Picador any minute now. So special thanks for making the time to join us today, and a big welcome to Professor Lisa Feldman Barrett.
Thank you so much. Thank you so much. So I want to start just by thanking Mark and Andy and everyone else at X-SPECT involved with the conference for inviting me to talk with you today. And I want to thank Giovanni for an absolutely spectacular talk, which is, you know, I'm very familiar with his papers, and they are on the regular reading list in my lab. But it was a brilliant talk, and actually there were some points that you were making that I was nodding so vigorously that I thought my head was going to pop off my shoulders. So, and I'll just say, it's a little daunting, you know, to give this talk, particularly to this audience with some folks here, including Andy, you know, who I've learned from, you know, very much. So this is for all the youngsters in the audience that, you know, that feeling when you're presenting to people who you really admire, that kind of jittery feeling never goes away. So I'll also apologize in advance because things here in the US are a little tumultuous, as I'm sure you're aware. And in my house, no one got any sleep last night or for the past couple of nights. So you'll forgive me if I have a bit of word finding issues this afternoon. So I'm going to launch the talk. Okay. All right. I decided to go really big today, and rather than dig into the details of various studies, really just to give you an overview of the particular approach to predictive processing that we take in the lab. I think that it's interesting to me that Giovanni ended with comments about his suspicions or concerns about psychologists, you know, sequestering off particular phenomena and then, you know, naming them as processes, all based on, you know, common sense, rather than looking for underlying computational mechanisms for an integrated brain. And what's interesting is that because this talk is only an hour long, I took out the first 4 slides, which were basically, I was walking through the history of psychology in one slide through a series of sequences to basically talk about how for the last 150 years, faculty psychology, which is what it's called, has really reigned in psychology and in neuroscience. And so instead, today, what I want to do is offer 3 hypotheses about how a human brain in constant conversation with a human body and the surrounding context creates all of the phenomena that we think of as being part of a human mind. So rather than assuming that we have types of memories and perceptions and emotions and actions, each with its own dedicated psychological process or mechanism, that instead, where each mechanism is dedicated, is implemented in you know, sets of neurons that are dedicated to that specific function. Instead, what I'm going to do is kind of sketch for you a common set of computational hypotheses that attempt to unify types of mental activity and actually integrate them with also what we know with evolutionary and developmental neuroscience. And so I'm really going to focus on, of the many things I could talk about today, I'm only going to focus on kind of 3 general points. And I should also say I'm kind of pitching this since I don't really know how familiar, I'm assuming some people are really familiar with predictive processing models and some people who aren't. I'm trying to pitch it right in the middle. So hopefully if there's any time for questions at the end, people who want to dig into some of the details that I'm not presenting today, I'd be really happy to discuss those. So the first hypothesis, I thought we could start with a doozy, something super unintuitive. And that's the idea that your brain constructs concepts. And what it's doing is constantly constructing concepts for the purposes of guiding action and constructing experience. And so in my work, instead of starting with folk psychology categories like memory or attention or vision or emotion, we usually start with the brain's perspective and we ask questions about how the brain functions and is structured. And then we go from there and then ask, okay, well, given that we have this kind of brain in conversation with our bodies and the world, how does that brain create the experiences and guide the actions that we have? And so I like to start with this insight, which is that your brain, basically for your whole life, is entombed in a dark, silent box called your skull. And it learns what is going on in the world only indirectly, from scraps of information that come in from your ears and eyes and other sense organs. And these scraps of information are the outcomes of some causal event that's happening in the world. And your brain has to figure out what caused these inputs so that it knows what to do about them to keep you alive and well. But there's a tricky bit, and that is that the sensory information is often noisy and ambiguous and incomplete. Any sound, like a loud crash, for example, could have many causes. A loud crash or a loud bang could be someone dropping a box. It could be someone hitting a car on the street. It could be, if you're in the United States, a gunshot. And in much the same way, your brain is also continuously taking in sense data from your body. For example, an ache in your gut could be hunger. It could be anxiety. It could be a gut feeling that someone is not to be trusted. An ache in your chest, for example, could be anxiety, or it could be the beginnings of a heart attack. And that the inferences that people make sometimes determine whether or not you live to the end of the day. So your brain has to determine the causes of sensory changes when all it has access to are the effects. And scientists and philosophers call this a reverse inference problem. How does your brain solve this reverse inference problem? How does it infer or guess at the causes of sense data when all it has access to are the effects? And the answer that scientists have, many scientists, including some people watching this talk, have determined is that by remembering past events that are similar to the present in some way. And so when the brain is faced with sense data, it's not asking the question, what is this? It's asking, what is this like? What is this like based on my past experience? So the last time I was in this physical state, surrounded by this physical array, sense data, what caused these sense data? What did I do next? And so on and so forth. Now, in psychology, a group of things that are similar in some way is called a category. And a representation of a category is a concept. So when your brain asks, what is this pattern of sense data similar to, you could say that it's constructing conceptual categories. Now, a category is not something that you have stored in your head, like a tidbit of knowledge that you can pull up and use whenever you need, like a file from a file drawer. A category is something that your brain makes as needed in specific situations. And I'm just going to walk through a toy example to show you what I mean. So here is a category of a group of things that are similar in some way, a category of cats. Cats are similar in various ways, right? They have whiskers, they have pointy ears, they purr. And for many years, scientists believed that this list of mental features was how you knew what a cat was, right? Does it have whiskers? Check. Does it have pointy ears? Check. Does it purr? Check. And so on and so forth. Yep, it's a cat. And this list was thought to be the concept of the category stored in your head so that you could retrieve it whenever you need it. But here's the thing. There's plenty of variation in this category. So some cats are large, other cats are small. Cats vary in their eye size and their eye shape. Cats vary in the color of their fur and the thickness of their fur and, whether they even have fur. So the simple list idea doesn't really work, which is called the classical view of concepts, doesn't really work in the real world. In the real world, your brain is representing concepts that are not defined by mere physical similarities. They're defined by something else, which is their function. So, for example, when I ask you to tell me the features of a cat and you're in a pet store and your goal is to have a pet, then your brain would construct an exemplar, an instance, and read off the features of the that would make for a good pet. If I ask you to tell me the features of a cat that you would see in a zoo, your brain might construct the instance of a tiger or a lion. And after you've seen a mouse in your kitchen, if the function of the cat is to catch a mouse, then your brain might, well, it might not, construct Bill the cat from the cartoon strip Bloom County, but it would construct an image of a cat with features that were specifically good for catching mice. And if I asked you to think about a cool cat, somebody who was really hip, you might conjure an image of the dude from The Big Lebowski, which is a film with this really cool guy in it. So notice that the various instances of cats here are all physically different, but they do share a function. And so the concept for cat changes with the situation because the function of cat manifests itself in different ways in these different situations. And you can take any kind of category that you think of as a concrete category and show that it's actually an abstract conceptual category, like a bird, for example. Whether what is the best exemplar or the prototype of a bird if your plan is to eat the bird versus to have a pet versus to see a bird in a park. The instances look physically different, but in a specific situation, they all share the same function. And this is how your brain, we think that your brain makes conceptual categories as you need them. So what's this? Well, it's a reverse inference problem. Many people at this point, when I ask what do you see, would say they see a cat. Why? Because your brain just made a concept for cats in the past series of slides. And so it's easy for you to see this as a cat. And we have a name for this in psychology. We call it priming. But of course, there is no cat here. And what if I told you instead that this is supposed to be an apple with a leaf on a stem? If you look at the image, you can see that. Most people can see it. And so this is an example of how your brain is using past experience to construct a concept to make sense of these lines. So you know what to do with this object, pet it or eat it. Another really interesting thing about concepts is that they have other operating principles. And I really loved Giovanni's discussion of the way that the brain is generatively making novel concepts, because that's what this GIF illustrates. And I should say I've only listed, one or two papers here by Larry Barcelo, but a lot of the work, the human work on the topic of conceptual categories, and particularly the way that the brain makes novel categories, really comes from his work. So your brain creates concepts creatively out of bits and pieces of past experience generatively, right? I'll wager that no one has actually ever seen in real life electrical towers playing jump rope before, but most people can immediately understand that that's what's happening in this image because your brain was able to combine bits and pieces of past experiences to create this really completely novel concept so that you can understand what you're seeing. And this illusion also demonstrates another really cool thing about concepts, and that is that some people can actually hear the tower hitting the ground. And some people can actually feel the thudding as the tower hits the ground. They can feel it in their chest. And if you can, that's because your brain, when it's making concepts, does this other really cool thing, and that is it's changing the firing of its own neurons to anticipate the sense data that it expects to receive. So the visual input is leading the brain to anticipate changes, somatosensory changes, and auditory changes. And this is the idea that concepts are not abstract, amodal representations, but in fact, they're quite modal. So it's a concept is a physical thing. And here's just a bit of data from one study just to sort of hit that point home. And so this is a brain imaging study that we conducted actually a number of years ago. So what you're looking at here on the left is a medial view of the brain, and on the right is a lateral view. The front of the brain, for those of you who are not familiar with brains, the front of the brain is on the left. The lateral view is sliced in a little bit so that you can see the underlying, you probably can't see my pointer, but yeah, I don't think you can see my pointer, but you can see the underlying, the insular cortex, which is buried deep in the Sylvian fissure underneath the temporal and frontal and parietal lobes. And so the blobs of activity here are thresholded changes in activity, in bold activity, during an fMRI study as participants are lying in the scanner. And what they've done, they've just heard a very short scenario where they've been asked to imagine in an embodied way this short scenario. Something like, you're driving home after a late night and you've had a little too much to drink, and you get really tired and your eyes start to close and you feel the steering wheel slip from your hand as you feel the tires bump, as they go over the safety guard on the road. And so after they've heard the scenario, subjects are asked to embody and imagine this scenario, and there are a number of scenarios like this. And what you see is really remarkable. So even though the subjects are lying completely still, stock still in the scanner, you see an increase in activity in motor cortex, premotor cortex, and somatosensory cortex. Even though their eyes are completely closed, you see an increase in activity in primary visual cortex and early visual cortex. And even though they're not moving at all, as I said, you also see an increase in activity in primary interoceptive cortex, which is considered to be the primary sensory region for sense data related to changes in your cardiovascular system, your respiratory system, your immune system, and all the other systems of your body. And the really cool thing is that you also see an increase in activity in a whole matrix of subcortical regions that are involved in controlling the body, in controlling the autonomic nervous system, the immune system, the endocrine system, and so on. So these are regions in the midbrain and the brainstem, which are thought to be important for what's called allostatic control of the body. Just by imagining, by being given a conceptual scenario and being asked to imagine it. So this is just a snippet of a small number of studies to show that psychological phenomena depend on a brain that continuously creates concepts. Now, you know, we have a lot of words for what the brain is doing when in the various instances or examples that I gave you. Memory, concepts, as you'll see, some people call them simulations, some people call them perceptual inferences, and you know, like Giovanni, I'm suggesting to you that really there's just a common set of processes here, and a lot of the phenomena that we think about in psychology as being very separate are actually can be understood under this same the umbrella of this this common set of processes. Now, the hypothesis, the second hypothesis that I'm going to get to is that your brain, in order to, as your brain is making concepts in an ad hoc way, in a situated way, to make sense of sense data coming from the body and from the world, that it's actually doing all of this predictively. Now, you know, giving a talk to a bunch of people who study prediction in order to, is a really daunting thing. There are a lot of details here, and what I've decided to do really is just, in a very gist-like way, lay out what we've laid out in great detail in a lot of our papers. And I'm doing this because in the third section, when we talk about the body, I'm going to talk specifically about the neuroanatomical substrates that support the hypotheses that we talk about in about concepts and their link to prediction. So the general idea here is that you're walking around, you know, with a brain that is loaded with bits and pieces of the past that your brain can re-implement in novel ways, right? So you have a generative model, and that generative model is predicting your future using concepts at some temporal span, the temporal span that is relevant for your species. So other animals do this too, relevant, you know, with the temporal span that's relevant to their species. For us, that can be, you know, a moment from now, you know, a week from now, or 35 years from now. But the idea here is that your brain is constantly predicting your future at various time scales, sometimes simultaneously, one of which will become your present. And scientists like to say, Giovanni said it too, that, you know, the brain runs a model for, you know, of the world. I think that the brain is running a model of your body in the world. And so from your brain's perspective, you know, your body is as much of a mystery as the rest of the world in you know, from this reverse inference sense. And so actually, as Giovanni was giving his talk, I was thinking, well, you know, sure, under certain circumstances, the generative model can become detached from the current context exteroceptively, but your brain can never become disconnected from the internal context of the body unless you have pharmacological help, right? Maybe with psilocybin or with ketamine. So a lot of what we think of as spontaneous cognition in the human, meaning that the mental events are not being driven by the outside world, fail to consider the fact that actually your brain is very likely to be continuing this conversation with your body. And so the idea here is that what we think of as predictive processing, which I don't have to explain to you what this means, but the idea of predictive processing is as important as for understanding and explaining the perceptual inferences that create vision and audition and so on, as much as our own sense of our own bodies. And I know Anil Seth is here and is watching this talk, and he's done some beautiful work on this topic. And I know that interoceptive predictions is one of the things that this group, the X-SPECT group, is really interested in. I sort of flash these papers to you as a way of just signifying the fact that I can't, and no one can really do justice to all the fantastic work that's being done to summarize all the evidence on predictive processing. Nonetheless, in my view, this work, which now I think my library totals over 600 papers, really suggests that the predictive processing approach forms a coherent neurobiologically inspired research program to explain how the variety of thoughts and feelings and perceptions and actions that we have are constructed using a common set of anatomical and neural features, the same general causes. And because of our focus not only on the perceptual inferences that the brain makes about the world, but also about the body, for us, our approach really attempts to collapse the artificial boundaries between cognitive, affective, and social phenomena, and really attempts to provide a common framework for understanding all the things that can go wrong in mental disorders, neurodegenerative disorders, and even physical disorders, that there's really a common framework to understand those in. And for us, it really starts with the idea that, your brain is a substantial metabolic investment. It all really comes back for us in the end to the metabolics of brain function and body function. And we've been spending quite a bit of time in my lab trying to understand that. But for today, I'll just say that, your brain basically takes up about 20% of your entire metabolic budget. And estimates from Mark Rakel's lab and others suggest that about 70% of the budget is spent on intrinsic activity, which we would think of as predictive activity. And that means basically about 14% of your metabolic budget on a daily basis is dedicated to running your brain's internal model of your body in the world. That's a lot. And so, evolution, to some extent, the importance of something evolutionarily, speaks with its metabolic feed. When something is metabolically important, usually it's because it's important functionally as well. And so here's a cartoon description of what we think is happening. Basically, your brain is continually talking to itself. It starts with initial conditions in the body and in the world, and then it projects itself forward in time, guessing what the body has to do next, and as a consequence, what you will feel and see and hear next. And these guesses begin as a population of past experiences, as a concept that your brain is reinstating or re-implementing in a generative way. These past experiences, this concept, are hypotheses about what is going to happen next. It's a plan for action in a specific situation for how to move your body, what the visceral and other internal bodily system changes are needed to support that action, which we call visceromotor. And also, as a consequence, what are you likely to see and hear and smell and even feel as a result? So these are what you would call perceptual inferences about incoming sense data. And so if you think about your brain as a statistical inference engine whose function is to guess at which actions are most likely to be beneficial in a situation and infer the probable sensory changes and the causes of those changes, then you could say that concepts are guesses about possible futures. they are candidates to explain what sense data will arrive to your brain in a given moment, just like you experienced with the example of a cat, or an apple. And sense data from the body and from the world help to select which exemplar of the concept that your brain just made best fits the situation. Now, the sense data from the body and from the world are not the only information that helps to select which representation will be completed and control your action and construct your experience. You also have an internal attentional system which also helps that selection process. And I'm not going to talk about that in this talk, although I'm really, really happy to talk about it in the question and answer period if there's time. But the idea is that this is happening really automatically, and that once a selection is made, we can say that the sense data are categorized, and the plan for action is then executed. And so this is what we mean when we say that sense data are categorized by a concept. And of course, each time a hypothesis is chosen, its prior probability is strengthened, making it more likely that the brain will construct it and use it again in the future. When the sense data don't match the concept that's made, or don't match any of the possible representations, then obviously we call that prediction error, and the brain has the opportunity to encode it. So I just want to go over this now one more time in a little bit more detail, just so that you can see the order in which our hypotheses have some like a hierarchical order that is guided by anatomy, essentially, and to some extent to the extent that we can test this temporally in data, is pretty consistent so far with a lot of what we've observed. And so it goes something like this. If you were to freeze time right now and start with the current state, your brain would be making a prediction about the next state. Or if you choose a dynamical systems language, you would say where the brain trajectory will take it in its state space. So you're projecting forward in time, constructing representations of what's going to occur in the next moment. These memories, the first thing they do is they prepare the body to act. So allostasis is the brain's continual regulation of the body. And so these are in a sense, visceromotor predictions, as well as motor predictions of what you physically are likely to do. And the hypothesis in R and other predictive coding approaches is that that your brain will make an efferent copy from which will be sent to sensory regions. So these would be collateral neurons, or collateral axons, I should say, off the main axon. And so taken together, this is a concept. The concept is both the motor and the visceromotor plan, as well as the perceptual inference. And the idea is that what your brain is doing is it's creating a set of hypotheses about the various things that could happen, which are similar to what's the present conditions based on past experience. So it's a sense of predictive categorization about what actions are needed, which will determine what your experience is. And notice in this case that we're not hypothesizing that the brain is evaluating something and then creating an action plan. It's basically creating an action plan, and that is the evaluation. It's not sequential in the way, like, say, appraisal theory proposes. And so the hypothesis is, like other predictive processing views, that data come in from the body and from the world. And if they verify or match the sensory simulations, then that releases the action plan, and that becomes your experience. Sometimes, however, oh, and then on you go to the next state. Sometimes, however, we think of that as automatic processing. Sometimes, however, the data don't match. And so this provides the brain with the opportunity to make a correction. There's a lot to say about how the brain might do this, which I'm happy to talk about afterwards. But I think the general point is that, you know, in psychology, we have a really fancy name for the brain taking in prediction error and correcting its predictions. We call it learning. That's what learning basically is, the encoding of information and the retention of that information so that the brain can use it later to predict. So the idea is that this changes the motor and visceromotor predictions, and also launches a correction to the prediction. And we think of this as controlled processing in psychology. So the thing I just want you to consider for a second is that most of our experiments in psychology and in neuroscience, when we're dealing with humans and even non-human animals, really assume that a brain works like this, that the brain is off, there's some stimulus, it's processed by the organism's brain, and then there's a response. And so it's like a relay race really in the brain. And we design our experiments with independent sequences of stimulus and response, effectively severing the contingencies between one moment and the next. But if your brain actually works like this, then you're basically taking this continually predicting brain and sticking it into an experiment where it's almost certainly going to be the case that it's wrong on many, many trials, forcing it into a mode that favors encoding and the prediction of prediction error, which is very, very stimulus driven. When in the real world, the dynamics may actually frequently favor prediction, except in non-neurotypical brains. And so you might say that standard randomized designs really encourage oversampling of what you might think of as being a less natural state of error processing. And this may be one reason why studies often fail to generalize when you go from the lab to the real world. So the main point here the main point of this section is just to set up what I'm going to say about the anatomy. But I guess what I want to say is that mainly, you know, we try to design not when I say we, I don't necessarily mean like you and me, but the larger field tends to still focus on stimulus and response when in fact, you know, any response is some combination of what's inside an animal's brain and the stimulus situation outside. And to some extent in the real world, what is even a stimulus is not dictated by the world, but it's dictated by the brain. And so this is really where the meat, I think, of this talk is, and that is the hypothesis that your body is at the center of your brain's predictions. That your brain is making concepts predictively because that's the cheapest, most efficient way to regulate a body. And the hypothesis really begins with an insight from evolutionary and developmental neuroscience, and that is that brains, all brains really on this planet, did not evolve for rationality or cognition or to be accurate in perception. Your brain's most important job is to regulate your body so that you can move it efficiently, and sensation and perception and experience all evolved in the service of regulating the body. Metabolic efficiency in regulating the body is a major constraint, selection. It's a major constraint or selection criteria for evolution. And physiologists and some neuroscientists really know this. This is a quote from Peter Sterling's book with Philip Laughlin. The core task of all brains is to regulate the organism's internal milieu by anticipating needs and preparing to satisfy them before they arise. And this is the definition of allostasis, the brain's attempt to anticipate the needs of the body in the future and meet those needs before they arise. And I'm pointing out that allostasis is not the same as homeostasis, and there is a current debate about whether or not the brain functions by homeostasis or by allostasis. From our perspective, we really come down on the side of allostasis as being the more useful and important concept here in how the brain is modeling the body. And what I'm going to show you is that that our way of thinking about predictive processing really places metabolism and energy regulation, as well as the sensory consequences of that regulation, which is called interoception, at the core of all mental activity. So in emotions for sure, but also when you think, when you perceive, when you act, when you decide, that representations of what you do are ultimately giving rise to what you see and hear and what you sense and what you feel, and even more generally, your sense of self, in part because of the central role of allostasis and interoception. And so by looking at the architecture, and I should just say that when we look at the architecture of the brain and we want to think about the dynamics of information flow, you know, there's a predictive architecture in the cerebral cortex. There's a predictive architecture in the hippocampus, as we heard Giovanni talk about a little bit. There's a predictive architecture in the cerebellum. I'm really going to focus primarily in my last few minutes on the cortex, although I'd be happy to talk about the relationship to the cerebellum and the hippocampus. And we're lucky in the sense that there's about 30 years of track tracing studies that tell us something about the architecture of cortical connections and the way that that architecture predicts information flow in the brain. So there's this very rich empirical body of work on primates and other mammals, which suggests that the arrangements of neurons within the cortical columns within the columns of the cerebral cortex predict information flow across the cortex. And that certain neurons, by virtue of their architectural arrangement and connectivity, will give rise to prediction signals that then propagate out to the motor and the sensory regions of the brain in the way that I described earlier. To demonstrate to you this literature, what I've done is I've taken Brodmann parcellations of the cerebral cortex. And I've used Brodmann parcellations because we could use the more recent and highly parcellated cortical maps, but that's not going to give us anything different than what I'm about to say. So I think using the Brodmann maps are fine. I'll explain. I'm going to sort of state the conclusion first, and then I will unpack it and explain it to you. That the regions that are colored in dark gray are the regions that are thought to be at the top of the predictive architecture or hierarchy in the cerebral cortex, where predictions originate in combination with the hippocampus. And these regions have a special name. And though that name is limbic. So the work from Helen Barbas and her lab and other labs suggests really clearly that limbic regions of the cortex are the most powerful predicting parts of the brain. And so there's a real irony here. And that is that for decades, scientists considered limbic regions to be the home of emotions, which are supposedly, your inner beast, this highly reactive part of your brain, which is really in need of control. But actually, the evidence suggests that limbic cortices give rise to prediction signals that drive action and perception throughout the rest of the brain, not just in emotion, but in all mental events. From the moment that you're born until the moment that you draw your last breath, the brain is predicting sensory inputs from the world and anticipating how to act on them, in large part guided by this suite or ensemble of regions. And it doesn't really matter whether psychologists or, individuals classify this as thoughts or feelings or perceptions or actions. It's really there's a single kind of cascade with a single set of processes that we can use. And so to explain this to you, what I'd like to do is to take the cerebral cortex and really, what I want to draw your attention to here is the little beige, ribbon of tissue that surrounds the internal parts of the brain. And this is your cerebral cortex. So if you take your cerebral cortex and you lift it off the rest of the brain and you stretch it out like a napkin and you look at it in cross section, you can see that the neurons are layered. They're arranged in layers. And so what I'm going to show you now is a cartoon version of a highly stylized cortical sheet, so that we can look at how the cytoarchitectural organization supports the hypothesis that the brain's internal model, its predictions, can be understood as a set of abstract conceptual categories or concepts that originate in limbic cortices. So the dark regions here are what are called limbic regions. They're, for those of you who know the lingo of anatomy, they are agranular regions. So these are regions that have very large pyramidal neurons, fewer neurons, and lots and lots and lots of connections. So I didn't draw the connections in here, but this basically, there's a lot of neuropil here, a lot of connectivity, and very few neurons. And at the other end, we can think of these white regions as, say, primary sensory cortices, like V1, say, where you have a lot of small neurons and many fewer connections. And so what's happening when a prediction is generated is that the neurons in the most agranular, most limbic part, when they send neural connections down to the thalamus and to the brainstem. These are the visceromotor and motor predictions. The collateral axons go to the upper layers of the next set of cortical columns, where there are slightly more neurons which are slightly less connected. And this continues as a cascade that goes all the way to primary sensory regions. So what's happening here is that probably in collaboration with the hippocampus, these regions like the cingulate cortex and the anterior part of the insula and entorhinal cortex are essentially taking multimodal, highly compressed summaries that's how memories are essentially reinstated in these regions, and inferring the sensory details in a cascade of processing steps so that the representations become more and more and more particularized in details until they reach the primary sensory areas. And so that's really what it means to say that, that the brain is generating a prediction. The concepts are not in the limbic regions. The entire cascade would be of information flow from the large pyramidal cells in these limbic regions to the smaller cells in the primary sensory regions. That whole thing would be considered the concept. And there would be not just one cascade, but multiple cascades with different priors. And then there would be some mechanisms to select which representation to use, including from the sensory periphery. So multimodal summaries become decompressed as they cascade from visceromotor and motor regions to sensory regions, generating perceptual inferences, which are inferences about the cause of sensations. And we can think about information flow for prediction error in the other direction. So what's happening, the hypothesis here, is that the information is being compressed, actually, so that the information goes from the multiple modalities, starting with highly particularized details. And the idea is that the redundancy is being compressed out of them, resulting in dimensionality reduction, by using successively fewer and fewer neurons to summarize the similarities until you get to the front of the brain in these limbic regions where you have these large multimodal, very sparse multimodal summaries, because it's very metabolically efficient to do this. And so compression, in a sense, is conceptual learning. It's taking in new information, but you're never learning that new information as a clean slate, even when you're a baby. And so that's the general hypothesis. And I should say, when I refer to this literature by Helen Barbas and others, that's literally what she finds. She finds that the cortical connections, that they're actually architectural arrangement, predicts information flow about 80% of the variance in information flow. Now, I'm just going to really briefly show you what this looks like in an actual human brain. So what we did is we looked to the macaque brain, and we identified particular regions that were these, limbic regions that we thought, based on Helen's and other people's work, that we thought to be the at the top of the predictive hierarchy, and where predictions were initiated. So, really important for the internal model of the body and the world. And so first we identified these regions, and then we verified, using track tracing studies, their connections to other parts of the brain. And then in humans, what we did is we identified the homologous regions in the human brain. So this would be the subgenual anterior cingulate, the pregenual anterior cingulate, mid cingulate, medial, and both the medial and the dorsal, actually that should say ventral, well, it's medial ventral anterior insula, and the dorsal ventral anterior insula, that should say lateral ventral anterior insula, sorry about that. And what we did using resting state data, so when subjects are lying in the scanner and they're not being probed by an external stimulus, we measure their blood flow changes with the Bold signal, and then we compute connectivity by looking at the mutual information in the signals across these areas. And that gives us a map. So here the blobs are not activity, they're correlations between activity in these regions. And so we had a bunch of these maps, and then we cluster analyzed these maps. And I should say, this was done on over 700 subjects. So we split the sample in half, and then used half as a discovery sample, and then replicated it on another half. And so this is the system in the brain that is important for seeding predictions. And for those of you who are aware of this literature, this system is made up of the system that is called the default mode network. It's also called the conceptualizing network, the mentalizing network, the self network, what else does it go by? The semantic concept network. And this network has more aliases than Sherlock Holmes. And the other network, which is well known, which is the salience network, which also has its own set of names, including the ventral attention network, the singular opercular network. Neuroscientists name networks basically in line with what they're interested in. And what we found, anyways, was that when you look at the connectivity between these cortical limbic regions and the rest of the brain, you find these two networks and where they overlap. And it turns out that these two networks overlap in a set of hubs, some of which are considered to be rich club hubs from the connectomics work. So these are considered to be regions that are densely connected with other regions of the brain, and important for as a backbone of communication across long distances. Now I want to point out that, and I should also say, we just replicated these findings in a 7 Tesla scanning environment with 100 subjects who were scanned multiple times. These are from a 3 Tesla, standard 3 Tesla scanner. What's really interesting is that primary interoceptive cortex is not only part of both the default mode and salience networks, but it's also a point of overlap. And what that means is, that interoception is always with you in every moment of your life, whether you're emotional or not, and whether you're aware of it or not. Interoception is not your awareness of your bodily state. It's the information about the body that the brain is using to model the state of the body. And it's very, very connected to these low dimensional representations, which, we call affect and the average person calls mood. Feeling pleasant, feeling unpleasant, feeling calm, feeling alert. So the hypothesis here would be that, interoception is with you every moment of your life, whether you're emotional or not. And so is affect. So affect is not emotion. Affect is a property of consciousness, because it's always with you. It's not specific to emotion. And just to finish up quickly, I'll show you that this system, which we seeded from these cortical limbic regions, have dense projections into the brainstem, areas that are, I'm just going to go really quickly through this. So now you're looking, I have the networks at the top, and then what I've done is I've shown you a coronal slice and a mid-sagittal slice now with the front of the brain on the right, and I've projected the connections into the volume of the brain so you can see the subcortical connections. And so both of these networks have extensive subcortical connections that overlap in a whole matrix of regions that are important for controlling the body, including the parabrachial nucleus, the periaqueductal gray, and a whole set of other regions. These two networks, which make up this system, also project directly to the bed nuclei for norepinephrine, for serotonin, and for dopamine. Now, norepinephrine is considered to be important for learning, and particularly learning prediction error. Serotonin and dopamine have been talked about in a number of ways, but what I want to point out is that evolutionarily, these regions actually, these neurotransmitters are, they're metabolic regulators. And so there's some really interesting things to say there about the links between regulating attention and metabolic function. And if you look in the brain imaging literature, what you will see is that the default mode network, amongst its many, the many things that it's been implicated in, are also semantic concepts. That is the dense multimodal summaries of concepts, including emotion concepts. Whereas the salience network is considered to be important for basically determining or predicting what sensory information is important to encode. Like, that's what salience is. So it's related to visual attention, to visual and auditory alertness. And in these studies that force the brain into a prediction error mode, where you're constantly presenting stimuli that are randomized so the brain can't anticipate. If you look at the hotspots in brain imaging studies across almost 6,000 studies, what you see is the increases in activity in the salience network meta-analytically, suggesting that this network, so if the default mode network really you could think of as the core of the brain's internal model of the body and the world, the salience network, we would hypothesize, is predicting the precision of incoming sense data from the body and the world. Basically, which prediction errors the brain is predicting which prediction errors it needs to learn in order to predict better in the future. I had more to show you about the relationship of certain gradients, so cortical gradients related to prediction, and the overlap of these cortical gradients with gradients that you can see in the hippocampus and in the cerebellum. but I think I've gotten too ambitious for the amount of time that I have. So I'm just going to skip that and say that the main insight here is that your body is part of your mind. You have a mind because you have a body. Your brain is always maintaining allostasis, and therefore it's always doing interoception. It's always modeling the body, and it's always creating concepts to make sense of your body, what's going on in your body in relation to what's going on around you in the world. And in this way, you know, mind and body are linked, not in some kind of gauzy, mystical way, but in a very, very computationally real way. And so the takeaway here is that, you know, the work that we do really uses predictive processing as a framework for understanding how a brain continually constructs concepts in the service of regulating the body. And that everything that you see and hear and feel and think and do is in the service of regulating the body, in addition to whatever else, you know, what other motivations that you want to assign to it. And that what predictive processing is doing is estimating the needs of the body, trying to predict the needs of the body for the purposes of action to maintain allostasis, which is balance. It's not trying to get to a zero state or a low state of energy consumption. It's trying to get to energy efficiency. And when the brain predicts correctly, that you could discuss that as categorization, which is guiding actions and becoming your experience. And so that's the hypothesis. That's the general hypothesis. In addition to my scientific papers, I've also discussed this in a popular science book on emotion. It's about emotion, but it's really largely more about the brain and the mind in general. And thank you, Andy, for mentioning my new book, which is coming out soon. And I want to thank my fantastic lab, who actually did all the heavy lifting here from the studies that I presented to you. And thank you for your attention.